Yeah, Bradley, hi.
I can't hear you very well.
Can you hear me?
Could you up the volume of your? Right. Mainak has to leave. He'll rejoin. Hello?
Can you get the sound now?
Yeah,
Can
I
you
can
hear
hear
me?
it. Yeah,
Okay. Okay, great.
good. So welcome to the meeting. You're the only one here right now, but you and I, that's okay. So how was your week?
It went well actually. It was pretty good.
How are you doing otherwise? Like how are things going?
Things are going pretty good actually. They are going way better than I had expected, and until now I didn't really hit a major hurdle. So they are going good.
Okay, good. So you have a report for this week?
Yeah.
Okay.
Yes. I'll share my screen then.
Yeah, I saw your Slack posts. I thought they were pretty good. The little videos you made. So good.
Yeah, I'll be sharing them too. So
Okay.
I'll be walking you over them. Okay. So just let me know if you can see my screen.
Okay.
Can you see my screen?
Yes.
Yeah, great. So I'll just go over this week's update. Okay. So you can see my screen, right? I'm just
Yeah.
trying to make sure.
Yeah, I can see.
Okay. So for week 7, I started out with I basically had to convert the nucleus segmentation model to this other format called ONNX, which stands for, I guess, Open Neural Network Exchange. So I've linked to the code here, the code that I had used to basically convert the PTH file to this format, to this other format. And then after that, I started to work on, I started to work from scratch again on the DevoLearn GUI. And this time I was using another library called Streamlit, which is very similar to Gradio, but it has more capabilities. So my goal was to have one standalone web app which would have every single package available in DevoWorm. Like it would be only one link for the home page, and then from there the user would have the option to go into the other use cases. Okay. So after that, I had to build the pipeline that was used to run inference using the Onyx models, and then I had to also run tests to ensure that the models really work from the GUI itself. And yeah, I already talked about this, I guess, because yeah, no, I did not. Okay. So my goal is to give the user the ability to select any of the models available in the library from the drop-down menu to the left, and then he would also be able to drag and drop the inputs. Plus, I've also included some of the examples in case the user doesn't have any example inputs with him. So I don't think this is very clear, so I think I should just open up the larger video. So can you see the video?
Let's see. Yeah, I think it works.
So this is the home page that I was talking about. This is the main home page from which the user would be able to navigate. And as of now, the home page basically contains some of the animations which showcase the capabilities of the library. But I'll be making changes and additions to this very soon. This is more like a placeholder just for now to see, like as a placeholder. That's all. So after that, okay, so that's the home page. I just scroll through it. Okay, so in the left we'll have a drop down menu in this format. So here we'll have the will have the listed models available in the library. So if the user selects this package, and then he will get the two examples here, and then if the user presses run, then the outputs would come up to the right. And as of now, this basically runs locally on my machine, but I'll be hosting them online very soon. I think I'll try to do it by next week.
Okay.
So this video is not working. Okay, so can you see this video?
Yeah, I think
This
so.
one?
Yeah.
Yeah. So this was another one of the models that I could integrate into the GUI. So here you can see like there's also the drag and drop functionality available. Like, just drag it here and you press run, and then you get the segmentation map to the right. So these are the two models that I could integrate until now into the GUI, and the rest of the two models I'll be doing it very soon. So we'll just go back to the blog post. Okay, so these GIFs, they basically show what I had shown in the video. So if anybody wants to go over them, they could just look at the GIFs. So
Okay.
And yeah, I already talked about the home page and the animations that are there. And yeah, so the work for this could be found in the other repository that I have, which contains the DevoLearn web test stuff. So because I'll be testing it in that repository, but then before the final hosting, I'll be shifting it to the official repository. So that's not an issue right now. Just for testing, I'll be using my own repository. That's all. So for next week, my goal is to basically finish building the GUI for DevoLearn, and then try to get it hosted online somewhere. And then I'll also try to use Plotly to have interactive plots here, because after the user gets the output to the right, it would be better if the user can also zoom into it and just get into the more details of it. You could just hover the cursor and you'll get the coordinate values, and you'll get the pixel values and all that stuff, in case he wants to get into the details. Plus the goal with this online platform is to basically act as a demo rather than to run inferences in a professional manner. I think that's the goal with it. But if it works well and if it works fast, then I guess it could also be used it could also be used as a replacement to the entire library. So that could also be expanded upon later. So yeah, so I'll be also looking into ways of running inference in bulk using the web GUI, and then moving from Travis CI to GitHub Actions has been a goal last week. I really couldn't get it done this week, but I tried to get it in as soon as possible.
Okay.
So that's it for this week's update. Thank you.
Well, that's good. Yeah, very good. So I wanted to go back over a couple things. You were showing the different models, and you could select.
Yeah.
So what are the different models that you have in there now?
Yeah, I'll just go ahead and open the video. So you're talking about this drag and drop.
Yeah, the drop
menu
down.
to the left.
Yeah.
So at this point, I only got the first two models to work using the GUI, the cell membrane segmentation and the nucleus segmentation, which are available in the library. So I got these two to work right now using the GUI, but the rest of the other two, I didn't get the time to integrate this into the GUI. So I'll be working on that soon.
Okay. Yeah, I just wanted to see what you had in there.
Yes.
So you have the cell membrane, you have do you pull them back up? Just the drop down.
Yeah.
Yeah. That's back. Yeah, there we go. The nucleus segmentation and the lineage populations and the embryo images are the ones that you have yet to get into the GUI.
Yes.
Okay, yeah, that's good. Yeah.
I'll be getting into the lineage population model very soon.
Okay, yeah.
So that would be another journey. So let's hope for the best.
All right, and then you have the Colab notebook and the Slack links in the.
Yeah. The home page contains that.
Okay.
The homepage basically contains the essential links that one could need. Like, they could have to start a notebook here, and the Slack link is here, so they could join it on the homepage itself.
All right.
So I could also add a link to the GitHub repository or the PyPy repository, so that could be done.
Okay. Yeah, I remember we have it released on PyPy.
Yeah, that has to be done. Yeah.
Okay. Yeah, good.
So I'm planning to have a new release by the end of GSoC this summer. So that's what I have planned.
Yeah. Yeah, we talked
I'll
about
get in touch with Mayuk and I'll just tell him when to have the new release.
Okay. That looks good. Do you have any pull requests that I need to be aware of?
Yeah, I actually have a pull request that you need to review.
Okay.
Just go to it right now. Yeah. So this GSoC 21 repo has a couple of has one single pull request that I made today itself. So
Okay, yeah.
so that has to be reviewed. That's all.
Yeah, I'll check it out.
There's a commit called upload on its module, so that has to be checked.
Oh, okay.
That's all. Yeah.
All right.
Yeah.
Sounds very good. All right.
Thanks.
So that looks pretty good.
Yeah.
Thank you for presenting that, and we'll be updating on that next week too. So we just keep going. And welcome Shruti and Jesse. Thank you for attending. Did Shruti or Jesse have anything to add? I know that Shruti just finished her NeuroMatch course.
Yeah.
So how was it?
It was fun. It was something very new, completely new for me. I'd never done something like this. A lot of brainy stuff. It was fun.
Good.
Yeah, I enjoyed it.
Yeah, and I saw your group's presentation on Friday.
Yeah. Yeah, I was like very happy to see someone I know among so many people. Like I was like, oh, Bradley's here, yeah.
Yeah.
Yeah, so that was fun. Actually, the collaboration and working together as a group, knowing so many people and so many different people from different places and their interests and backgrounds was amazing.
Yeah. That's good. And they're going to do the deep learning course in a couple weeks, I guess. And
next week, actually.
Next week?
From 2nd of August, yeah. I'll probably be an observer there. I'm not enrolled like a full time there, so.
Yeah. Yeah, and I know that Krishna's TAing that.
Yeah, he is.
And I think Jesse's attending that.
I'm going to try to do that because I'll be just moving to Boston at the time and everything else that's going on in my life. But I'm going to try to do that. I really wonder, I don't really get to pay attention as much to the computation or science year. Because last year it was just very intense. It was. I wonder if they modified the pacing of anything this year because there was a lot of different the ways the groups interacted, and there was a bunch of efforts afterwards to have like slow pods to go over the material again, and that may happen again this year because there's just a lot of stuff. It was a wonderful experience, and I'm very happy to do it. But I'm about to catch up with Shruti later about that. And I'm definitely going to try to do.
No, actually, they were saying that as compared to last year, this time they tried to give some extra hours for projects because last time it was just theory and there was so we have a whole day for project, like a special day without tutorials. So that was I think something different. The rest it was fun and nice.
Yeah, that sounds like a little bit.
It was hectic. And also it was very hectic, I must say.
Yeah.
By the end of the day, you were tired and you could not do anything else.
Yeah. So, I'm going to hope to do like the deep learning version of that, but I may be sliding the observer track. We'll see what happens. Yeah.
Hello, Sanjay. Thank you, Jesse. Yeah, it's I had two groups that I mentored, and then one of them, of course, was in Shruti's group where they were presenting and they were doing some work with fMRI, And then the other group I mentored was doing some work mapping between deep learning algorithms, deep learning models, and the brain, and some neural data from the brain. So they were looking at fMRI data and the AlexNet. And they were looking at how the different layers of AlexNet correspond to the different parts of the visual stream. And it turns out that there are a couple groups doing a similar thing. So there are a couple groups that took different approaches to that same type of question. And it was really interesting to watch, like, some of their results. Some people were looking, some groups were looking at, like, the nature of the input data and how that corresponds to what's going on in the brain, and then those activations, what's going on in a deep net. So you could, they have a type of analysis called representational similarity analysis, and that's what they were typically using to analyze the data. So you just take each level of the deep net, you take each part of the brain, each, like, they're, what they call regions of interest, and then you just do a bunch of correlations to see where there's more correlation in the output. So, like, the correlations are pretty low. They don't usually ever get above, like, 0.35, which is not great for a correlation, but, you can see differences in the correlations. You have, like, some relatively high correlations in some parts of the brain, relatively low correlations in other parts of the brain. And that's supposed to mean that they're doing similar things, perhaps, or that the processing is a similar way of processing information. We don't really know, but we know that, like, there's some interesting relationship there. And so I thought that was really interesting. And then, there's so many different deep net models out there. It's interesting to see how the results varied between the different models. So you have AlexNet, and you have, I can't remember some of the other ones that they tested, but there are a number of different models that were tested, and they all had a little bit different results. So I don't know if that's just because that's just, stochastic, or whether it's like there's something going on with specific architectures that, maybe we can learn from.
Probably the number of layers or the filters they use in each layer that cause the difference, probably.
Yeah, I think there's definitely keep your eye on that. I think there are going to be a lot of interesting results in that area where people are just mapping between the, brain data and deep nets. So, all right. So, Sanjay, did you have anything to report this week, or any news?
I didn't do anything actually this week. I was not ill, so.
Okay. Yeah. So, let's see. Today we're going to talk about, I wanted to go through some things. We'll talk about some papers, talk about, I've been promising to do this for a while, but I'm going to talk about, and this is something I do with my other group, how we can take computational agents and conceive of them as like a series of germ layers. So in this group, we talk about development, we talk about developmental biology, and we talk about the embryo. And one of the things we know about at least mammalian embryos is that most embryos, C. elegans has this, but we don't really see it very strongly, is our germ layers. And so you have 3 germ layers. In some embryos, you have 2 germ layers, but they have this sort of basic set of cells. They start out at different, like, so you have, for example, in C. elegans, you have, the 2-cell stage, and then the 4-cell stage, and the 8-cell stage. And when you get to the 8-cell stage, you have these cells that are going to become part of the body. So they're going to become like the skin and the organs and the brain. And then you have this other one cell that gets produced at the 8-cell stage. It's going to become the germline. And from that point, the germline remains sort of its own thing. So the germline is out here, and the germline produces cells, and it's eventually going to produce eggs and sperm. In the hermaphrodite's case, it's going to have both eggs and sperm, and it's going to produce eggs that they lay, and then they become new worms. But that germline is, segregated pretty early in development. and it just stays there. And there's a reason for that, and the reason is because you want to protect the germ line because you don't want to have too many mutations in it. For example, random mutations. You don't want to have the chance that the germline differentiates into something and then you can't make offspring. So it's, there are the reasons for that. But I'm going to talk about like how we might be able to take the germ layer model and improve sort of our way of thinking about computational agents. So, and then I'll probably go over some submissions. We don't really have much in the way of submissions to talk about, although I want to go over it once more. So I'm going to share my screen. All right. So the first we'll start here at the submissions document. Everyone can see my screen? Okay.
Yeah, we can.
Okay. So these are our submissions that we've been making. We revisit this every week. We haven't made don't have a lot of additions this week, but I did want to go over some of the things that we have outstanding. We have the DevoLearn paper. That'll be something after we get the new release out. The boring billion, is that going to be a book? Is that going to be something else? A paper? And in the boring billion category, I put a lot of the stuff that we've been talking about very early embryos from like maybe 1 billion years ago. And so that's something that is sort of an open-ended project. It could be a paper, it could be a book. There are a lot of research questions there to talk about. Then there's this Kindle book, which is this idea that Krishna had about bundling a lot of the educational materials into a Kindle book to put on Amazon. We haven't really touched that in a while. The Mathematics of DevoWorm, which is this poster that would become a book chapter or something similar, where we have all of the major equations of DevoWorm. And maybe next week I'll try to bring that back together, and maybe we'll try to see if we can add to it or if there are things that we need to change or what. But I think we have 4 or 5 categories. And, they may be equations, they may be like computational models. So it's just a way for people to see what kinds of things we're thinking about in terms of mathematical models. And so, you have deep learning models, you have connectomes, you have morphogenesis models, you have other types of things that are, have fundamentally different ways of being, fundamentally different representations of development. So, we can go through that and see what we can add or refine on that. And then eventually I would like to make that into maybe like a short chapter, or even like a technical paper. I don't know if we could make it into a preprint, maybe. It depends on how much we put into it, because preprints typically are maybe about 10 pages at least. So that opportunity exists. We have this Williamson symbiosis test, and this is using molecular data. Last week I talked about I talked about metamorphosis, and I had some articles that I showed people on the transition from a caterpillar to a butterfly. And so that's something that, you see in the phenotype, but the question is what happens in at the genetic level? Are there 2 programs that are sort of stuck together, or do you have this program that kind of just, transitions from one developmental pattern to another? And that's what we would be testing with that. There are molecular level simulations of diatoms. There are these morphological simulations of diatoms. There are these archaea, models of archaea, where we have these archaea bacteria, which form these different shaped phenotypes. They're single cell organisms. And then the idea would be to analyze them with some sort of approach. We talked about networks, where these networks that I proposed, which are these Euler path networks, different ways of doing that. There's this Eye of Nature book, Steve McGrew's magnum opus, I guess you could call it, and it's really an interesting book. He brings up a lot of topics in it, and he actually, in reading through it, he does hit upon a lot of these themes about like non-neuronal cognition and other things, and tying it to evolution. So it's a really interesting book. And I don't know, I guess there's a desire to like make it, sort of publish it posthumously, but that's something that I don't know, like, who would be interested in that. We might publish it as a series of blog posts or novelizations, and there are different ways we can do that. We're not a publishing organization, so I don't know, but definitely it's there, and and then there's this, and this is something I'm going to talk about today, follow up on today with this talk about the agents as germ layers, and this is differentiation trees of the brain. So this is something that, we look at brains and we say, how can we make a map of how the brain develops in terms of like, cell differentiation, what parts of the brain get there first, what parts of the brain get there next, and so forth. And so there's an article, we talked about my other group, there's some other possible ways of doing this, and I'm going to kind of get into that a little bit later. So those are our outstanding things that we've been going over for many weeks, and kind of just give people a reminder of what we're doing. No deadlines on the horizon. I think we don't really have any deadlines. There is this Dynamics Days Europe. This is something that I'm doing called Game Theory of Developmental Processes here. I changed the name of it. But it's a work in progress. So this is something I'll probably present to the group. This is due, I think, at the end of this coming month. So this is going to be a talk. And this is Dynamics Days Europe. So this is Dynamics Days. It's sort of a physics and complexity conference. They do a lot of people like from the dynamical systems community are there, and there are a lot of mathematicians and physicists and some computational biologists. And this group, they've hosted it every year in like Europe and the United States, and with COVID they've kind of had to move it online, and so the European group has been active in doing that. So this is why I'm presenting to the European group. I actually got the program already, and this talk is actually in a pretty interesting group. It's like a games and development group. So there are like 6 lectures in this series, and they're all different topics around it. They kind of synthesize the group based on like some of the submissions that they had. So it's actually, I'll bring it up when I give the presentation. It's a pretty interesting and eclectic mix of talks, and hopefully it'll be recorded online so we can see it later. So if we have deadlines, please let me know. I can add them on here. People want to submit things. I know there are things coming up. The NeurIPS workshops, for example, which are, for machine learning, for deep learning, those are coming up. So the main conference is actually quite competitive, but the workshops sometimes can offer like, subtopics, and those are, maybe less competitive. And they, you just write up a paper, you write up an abstract, they tell you whether you're accepted or not. And then you can give a presentation in one of these workshops. So, that's a good opportunity to sort of get involved with NeurIPS. And then, they'll be advertising these. So I think maybe more towards fall we'll find out what they are. But maybe there'll be some that are relevant to our group. All right, so this is our task board. I didn't really want to go over. I went over this last week, I think, pretty deeply. We have, again, our finished category. We have some things that have fallen off the radar. I updated this after last meeting. So there are a bunch of things that are kind of off the radar. But, those are things that, so if you're interested in the group meetings, task board, let me go in there, put that in there. And so this is for major tasks for this season, 2020-21. And we have a number of projects that are sort of, this is aside from the papers and the submissions. The papers and submissions are pretty focused, but some of these other projects are a little bit more broad. We have things that, you know, I don't know if people want to add things in or, but yeah, we'll be updating this later. I think I moved some things in different categories, and so. All right, so why don't we move on to, okay, that's DevoLearn, and so I don't think there's anything to update on that. So this is the thing I'm going to talk about. This idea of the vehicle. So this is something that, like I said, I work on with my other group, and we have this thing, and I'll talk about it. They're called developmental Bradenberg vehicles. So let me put this up here. So these are computational agents that they're not like C. elegans necessarily, but they're very similar in that they have these very minimal nervous systems. And so in a typical Bradenberg vehicle, you have this vehicle, which is this embodied agent, and it has a body and it has sensors at the front and effectors at the back, and you can configure the sensors and effectors any way you want. But the critical point is that the sensors and effectors are linked somehow. So in the classical Bradenberg vehicles, they're linked to the straight, straight connection, sensor to effector. And then you can have combinations from like the left sensor to the right wheel, or the right sensor to the left wheel, or you can have these, what they call contralateral connections, or you can have like a ipsilateral connection, which is the left sensor to the left wheel. That's just borrowing terminology from the brain. It crosses this midline, but notice that all the terminology is around this body, and so there's this set of spatial relations that you have with the agent. So one of the things we're interested in developmental Bradenberg vehicles, and this is the preprint where we have, this is something that we published I think a year and a half ago. This was some work done in conjunction with Google Summer of Code. So this is Bradenberg vehicles as developmental neurosimulation. This paper kind of gives a lot of the sort of the vision of it and some examples. So this is something that you might want to check out if you're interested in this. But the idea here in developmental Bradenberg vehicles, and Bradenberg was this, he was this cognitive scientist who used this as like a thought experiment. And since his time, people have built these things in a computer and robots, and they've been able to show that, like, even with very simple nervous systems, they can achieve some really interesting behaviors with respect to light stimuli or respect to other things. And so in developmental Bradenberg vehicles, we're taking it from what Bradenberg envisioned, which was a static phenotype with a static nervous system, very simple nervous system, and we're adding two things. We're adding this morphogenesis, so we're adding the ability to add and subtract neurons and connections, and also this sort of, so we have morphogenesis, and then we have this more complex nervous system, where you have not just these singular connections between sensor and effector, but these intermediate, or what we might call interneurons, which actually allow you to mediate some of the signals or attenuate them. So you have some sensors, or you have three sensors at the front of this vehicle. They're coming into this single interneuron, but there are also two of them are also mapping to this interneuron, and then these two interneurons are mapping to this middle interneuron here, and then back down to this one out here, which then goes to the wheels. So you have a multiple, and then this one kind of goes from the first interneuron out to the wheel directly. So you have all these layers of control that you can use, and they develop in development, so they give you these developmental stages that you can work on, and you can develop behaviors, and then hard code them into the agent. Now, where does development come in? So development comes in at the morphogenesis level, but also we want to get a little bit more detailed. We want to say, okay, we can exploit this idea of tripoblastic development. So we can take the body and the brain and think of it as a single sort of developmental system. So I mentioned before that you have these dipoblastic systems and tripoblastic systems. So in the in different in the tree of life, you have these ctenophora, which show like these dipoblastic phenotypes, and then you have Cnidaria, also have a dipoblastic phenotype, and then you have this evolution of something called the mesoderm, which you only see in tripoblasts or bilateria. And bilateria are, of course, us, C. elegans, and a number of other a lot of different organisms. And they have bilateria are defined by their bilateral symmetry. And so, briefly, the ectoderm is this sort of outer layer, the endoderm is this middle layer, and in dipoblasts, they just simply have a gut, which is like where the food goes in and gets extracted for energy. In the tripoblast, you have this gut, but you have the endoderm, which is here, the mesoderm, and the ectoderm. And these are going to become different parts of the body. Like I said, the mesoderm will become like, muscle. The ectoderm will have a role, and the endoderm will have its own role. And the way this is in the embryo, so these layers emerge in the embryo, not the early embryo so much, but like later on. And, they'll become different types of tissue. And sometimes this phenotype will be conserved so that you'll have like the gut will remain in the middle or at the midline of the organism. And then you have these tissues that sort of emerge around it. And sometimes they move around and become different tissues, but they maintain the same layering. So we use this as an inspiration for these agents. We start with this sort of tripoblastic origin point. It's just some concentric circles. And then from there we can take like this black inner part. We kind of play with the metaphor a little bit. That black inner part will then become like the sensors and effectors. So they kind of can move around to the edges of the agent. You have this outer layer, this outer shell, which remains throughout development. And then this middle layer, which in this case is going to become our neural network. So we have these cells or the space that it can become the neural network. And then wherever this moves will define where the network evolves. These sensors and effectors will position. So the idea would be you could develop an agent in the following way. You could define where these sensors and effectors, which are the inner layer, are going to be along the edge or maybe even inside the agent. You could define where your neural network is going to be. Usually it would be in the center, but it could be maybe on the outside edge. It's possible, If you're thinking about, like we talked about Hydra, we've talked about Hydra before in this group, and they're a marine organism that has this thing called a nervous net. And the nervous net is something where they experience the sensory world not through like this formal set of sensors and effectors, but through things that propagate through the organism. So they sense things, and then instead of having a central nervous system, everything just propagates through to different parts of the body. And so their neural networks are more integrated with most of their body than other organisms. And so this is, or at least like, it's not centralized. So you have this, you can put, and then this is the outer body here. So this would be the analogy of sort of the part of the Bratenberg vehicle that we didn't really talk about, which was the body. And so this is the body. And so we're moving from this vehicle metaphor to something more developmental. Now, in terms of the way this works in development, in terms of what we might call a differentiation tree, we can draw this tree of the different germ layers. And so I'm not giving too much detail here because I don't want to, I didn't really map this to the tree, but I just wanted to show how this works. So you have these different germ layers, and we'll call them A, B, and C. And so the branch length is important because the longer the branches are, the more chance you have for cell proliferation. So you can have like a bigger layer. So this layer here is a certain size. It can grow in size by having a longer period of cell division and proliferation. And so that can be mapped to this tree. And, this is something you can use, think about this as a computational mechanism where you're looking at, I can design a tree of this topology with branch lengths of a certain length, and then that would translate into a certain developmental vehicle. So you would go from this basic tripoblastic arrangement, implement this tree. It gives instructions on how this thing will look at the end. And the positions, it can, that's something different, a little bit different, but that's, that's not, the only thing. But this is basically the way that the different layers are differentiating and evolving. So there's this A, this B, and so in layer B, we actually see that there's this initial layer that happens, and then there's a split. So this B would be roughly analogous to these sensors and effectors, where you go from having one black blob to two black blobs that divide, and then they migrate. And that's, of course, not within the scope of this tree, but you have these two B layers, a B prime and a B prime prime, that then you can, those have independent instructions on how those will proliferate and when they'll terminate. And then C is this body, which then is roughly the same size, but again, it can proliferate for a longer or shorter time, depending on the length of the branch. So this is the way we think about differentiation trees. You have these trees that, like, basically when you start off with, like, say, a single germ layer, in this case, we actually are doing a little bit different than this model, where we have A and B. It's like A and then this BC precursor that starts out. So you would have a single, and I didn't show the single sort of germ, the sort of the single cell or, 4-cell case where there's no real differentiation of these three germ layers. But once you start to, you can start to differentiate these three germ layers using this sort of tree. You have A, and then you have a BC precursor, so that would be like the neural network, and then the body and sensor effectors would branch off. And then eventually you'd get the sensor effectors in B, and they would branch off from the body C, and then they'd become different things. And then eventually you'd get a mature vehicle, which would have all these components, but they'd originate in development at different points so that you could control, say, like the size or the position or the shape, the sort of the duration of time in which they're developing in proximity to one another. So there are all these things we can do with development. Then this is a slide from a recent talk on don't worry about the offloading part so much, but this is basically what it would look like. The agent phenotype, you have this model of innateness, which would be some sort of algorithm that would determine what this looks like from, say, an undifferentiated sphere to this. And then you'd have where agent would interact with its environment, and it would gain information that way. And this allows it to become a mature agent with a nervous system, and then training the nervous system. And then this is sort of the progression. You go from this three germ layered precursor to something that has different layers. The layers emerge over time, and then you end up with this vehicle. I know that I'm skipping a lot of steps in this, but this is basically the schematic here. And this is sort of reminiscent of some of the stuff that they were doing with the original, what they call animats, which were these agents that they would build like little robotic models, but they were supposed to mimic animals. So there were some that would mimic, like, rats in a maze or other types of organisms that were doing things, sort of intelligent behavior, and they'd model it using this sort of, some sort of little vehicle or little agent that they could control with a remote control. This is Norbert Wiener, and he built one of these animats. And so this is sort of the inspiration for this, among other things. So further developments on this, we still have to determine sort of what is the innate component that enables all this. So this requires you to have this innate component that tells the agent kind of what to look like. And so there are a number of options. One is a generative model with an unspecified internal representation. That means that you're generating things with an algorithm, and it's sort of, being evaluated at every step. So you don't have to give specific instructions on what it should look like. There's less control in something like that, but you get more interesting options. So that connected to that is sort of using genetic algorithms, and we've designed some genetic algorithms for this, but we really haven't been able to specialize any. And this would search for alternative configurations. So for the nervous system here, we've worked with some genetic algorithms to try to shape what this looks like and select the best topologies for this internal nervous system. But you can go further and say, what should this vehicle look like in development? You know, what kinds of things are developmentally advantageous? What kinds of vehicles are sort of the optimal phenotype for different tasks? So you can, you know, have a fitness function that says, I want to do this type of task with the agent. What's the best phenotype we can evolve or develop? And so you might get something that looks like this or something that looks very different. You might get something with a nervous system on the outside, for example, and a body in the inside that gives it rigidity. And, you know, there are different ways that can happen. We can also then use genetic regulatory networks. And I know we talked a little bit about these, but this is something that's separate from a genetic algorithm, and it's a way to sort of model how genes are regulated and their expression. And so these genetic regulatory networks usually use like some sort of input. They use a signal, and they, like, if you have a genetic algorithm and you have things organized as genes, and they get, you know, they get selected for, they get expressed, you know, genetic regulatory networks can shut them down or turn them on at different times. And so this is kind of useful for tuning outputs of a deterministic program or even something more like a genetic algorithm. And then finally, you can have hybrid programs which, you know, combine like a deterministic program with a genetic algorithm. And so you can see that that's very useful in a case like this where you're trying to map the differentiation of these different parts of the agent body, and then you want to figure out, you know, positioning of these things. So that would be, you know, maybe a different type of algorithm altogether. And so this is sort of the thing, and I'm presenting this because I wanted to, shout out for Norbert, yes. I wanted to, you know, hear if people thought about it. I don't know if people were interested in this group so much, but I also wanted to talk about it in the context of differentiation trees, because we talk about that a lot in this group. And so this is something that's ongoing in my other group, and we're kind of working out the details.
Yeah, I think this looks nice. I mean, this looks different from. It's nice and different. I guess I'll have to look into more about differentiation tree and see if I can understand more stuff about it and can maybe talk about it.
Yeah, I'm going to add to these slides. I just kind of gave a very quick overview of it, but we're preparing a paper for like an artificial life special issue. And this is something we've been doing on embodied intelligence. And so this is something we're going to be working out for that paper too. So it's sort of a time when like we need to have like in our paper, we already have the paper written, but we need to revise it. And we need like more specific things to put in it. You know, I don't know if we need to have like a lot of like established results, but we need to have like some things worked out and, you know, something to give the community for them to like be impressed. So it's like a matter of kind of figuring out what works and maybe what doesn't or what's plausible. But definitely, yeah, it's not so, I mean, people have done this sort of thing with developmental agents before with embryos, and they've used things like cellular automata to define the shape. But like, you know, saying that, you know, cellular automata usually just kind of give you interesting shapes. And so there's it's good because you can see what's plausible, or, you have a common set of rules and you say, well, what emerges if you just have a common set of rules? In this case, you have like maybe, your different germ layers have maybe different sets of rules, or you're thinking about it in terms of like, you're going from this blob to things with different functions, and then you're getting this agent that does, maybe has a behavioral aspect, but also has like this body and, other things that go along with it. So different objectives, I guess. So they do a multi-objective optimization, but that's very hard to implement. And so this might be a backdoor in some ways to that, for a very specific thing.
Yeah, sounds cool.
Yeah. Anyone else have any comments or? Let me see. Okay. Well, okay. So I'm going to go through our papers now, and I'm going to, as if you've been following along in the last couple weeks, we've been talking about environment, and this is what is environment 3. So this is last couple of times we've talked about light, and we've talked about, I think, thermal optimization. And again, to refresh our memory about this open question about environment. And the questions are, what is it? What is its relevance? And how do you define it? So this came out of a discussion we had about some papers where there's this term environment, it's very loose. And we talked a little bit about environment in the last set of slides about, like, there's this environment that's giving, influencing development. But what does that mean? And so we've come down with two sort of things about environment. One is that it's a permissive thing, so it's general information in the form of changes, stresses, and intensities that are transduced into the biological system. And two is that environment is instructive, and that's that specific information, ratios, patterns, and codes are
You're not sharing your
transduced.
screen, Bradley.
Oh, okay, sorry. I must have shut it off. All right, there we go. All right, so we're back to this document. So the first one is environment is permissive, which is general information is transduced into the biological system, or specific information, ratios, patterns, and codes are transduced in the biological system. And so those of you who've been at Neuromatch know something about, like, how maybe ratios, patterns, and codes are in the nervous system, and how maybe you have things in the environment, like, the position or the length of days versus nights. So you have things that trigger circadian rhythms. You have, rates of, like, sounds in the environment that are used by birds. Their song has, like, a sort of this coding or this ratio organization. So that those are things that, but then this general information, these stresses or intensities, these are, again, things like temperature or other things that we can quantify, or at least get our hands around so we can quantify it, or things like colors. So if we go back to this, what is environment? They have this paper, and this is actually in C. elegans. This is Eduardo Izquierdo is from the OpenWorm group, and there are a couple other people in this paper. And the title is Persistent Thermal Input Controls Steering Behavior in C. elegans. So C. elegans moves around with these very stereotypical movements, and they have these steering behaviors. So they do these loops, or they loop back around if they're moving in a certain direction. They call these omega turns, which kind of move in an omega pattern. And there are different ways that they can move around, and they steer their behavior towards resources or away from predators or things like that. And so this is an important type of movement for them. So the abstract says motile organisms actively detect environmental signals and migrate to preferable environments. So they're looking for things that they want to exploit, or maybe they want to avoid. And this is not just C. elegans, but even like in the Bradenburg vehicles, they do the same sort of thing. Especially small-sized animals convert subtle differences in sensory input into orientation behavioral output for directly steering towards a destination. So they're actually, I know we've talked about we've talked about psychophysics before. One of the things about psychophysics is that you have this ability to sort of calculate differences between stimuli, or between a background of noise and a stimulus. And so what they're doing is they're, like, looking at this difference between, say, like background noise and some signal, or two different signals, and they're able to integrate that so that they can move to one or the other. They can make a decision. And so there's this whole set, there's these kind of law-like behaviors that you can observe that are based on this idea of detecting these differences. So this is something that you see in a lot of different organisms, and C. elegans exhibits this. So they're able to steer their body towards the signal that they prefer, they can detect. So this is something that you'll see, but the neural mechanisms underlying steering behavior remain elusive. Here we analyze a C. elegans thermotactic behavior. So thermotaxis is where the organism moves towards a thermal source. So like, if you think about like a heat source, it might move towards the heat source. Or if it's too hot, it might move away towards the heat source. It models the environment as sort of this gradient, and it moves towards a certain thermal regime. Okay? So these are just very simple movements, and they're moving towards or away from things. So if you were to move the thermal source around, the movement would just move around. They'd start moving around towards wherever the thermal source was moving. And so, and again, when I showed the slides on Bradenburg vehicles, I showed the sun. So Bradenburg vehicles will move towards the light. They exhibit these taxis, these what they call phototaxis. In this case, we're talking about thermotaxis, so it's very similar. Which a small number of neurons are shown to mediate steering towards a destination temperature. So this is a very small number of neurons in the C. elegans nervous system. So we constructed a neuroanatomical model and used an evolutionary algorithm to find configurations of the model that reproduce empirical thermotactic behavior. We find that in all the evolved models, steering rates are modulated by persistent thermal signals sensed through forward locomotion. Persistent temperature increment lessens steering rates, resulting in straight movements of model worms, whereas temperature decrement, which is decrease in temperature, enlarges steering rates, resulting in curvy movement. So they're moving with respect to the temperature source, but they're also moving kind of maybe to sort of, they kind of, I don't know if they're anticipating it, but their overall movement in this, as they're moving it around or putting it in the same place, it's kind of affecting how they're doing this. Maybe they're just kind of moving and they kind of learn, well, you have to move a lot. You have to be like, you have to maintain this sort of place where you can move around, you can prepare to move. So persistent temperature increment lessens steering rates, resulting in straight movement. Okay. The relationship between temperature change and steering rates reproduces the empirical thermotactic migration up thermal gradients and steering bias towards higher temperature. Further, spectral decomposition of neural activities show that thermal signals are transmitted from a sensory neuron to motor neurons on the longer time scale than sinusoidal locomotion. So they actually are doing this in their nervous system where they're sort of integrating the information over multiple moves, and they're sort of observing it more long term than just kind of like a reaction. But the behaviors kind of show themselves as a reaction. This is really interesting. Our results suggest that employments of persistent sensory signals enable small-sized animals to steer towards the destination in natural environments with variable, noisy, and subtle cues. So this is, again, like they're using this temperature gradient and they're observing these different types of movement. And then, of course, they're looking at the neuroanatomical wiring, and they're seeing that there's this longer-term sort of activation within that circuit that shows maybe a little bit of anticipation. So they're kind of going. So there isn't a lot to this paper. It's a couple, I don't know if they have the figures are at the bottom. This is the evaluation of the behavior. They have this gradient, and then they show how this works. And then they show the neural circuit. So we're talking about 6 neurons, I think, or 7 neurons. So we have these networks where you have, a very small network of neurons, and they're controlling the steering behavior. And it's basically like, in sensory input, turn right, turn left, maybe reverse course, maybe, it's very simple. And we can distill this in C. elegans down to about 6 neurons. And so they show sort of the time scale of this. So they monitor the current state at every 0.1 second. There's this potential for a move. So they can make these different types of turns or curves. And then they exit that curve and they make another curve. And the nature of the body of C. elegans is such that they can make like an omega turn or a shallow turn. They can't just move in any direction or any orientation. They have to sort of stick to these sort of stereotypical curves. And And so they kind of go through this. Neuroanatomical models reproduce thermotactic behavior, so they're able to actually model this with an evolutionary algorithm or a genetic algorithm, and they're able to look at the circuit and how it's activated, and then they're able to they have the experiment and the simulation, and they match up pretty well. And so that's it. And then, you know, there are different temperature change regimes, so you just look at how that affects the curving rates. So this is a good example, I think, and it ties in nicely with this example from the developmental brain vehicle of how thermal inputs can, you know, drive behavior and drive maybe even development of nervous systems. So that's what is environment. Let's see. This is a paper where we're talking about genetic regulatory networks. And this paper it's a new paper in Cells and Development, and it's about cell regulatory networks. So the cell in the age of genomic revolution, cell regulatory networks. And so this paper is, the abstract says, over the last few years, an intense activity in the areas of advanced microscopy and quantitative cell biology have put the focus on the morphogenetic events that shape embryos. So that's what we're interested in with the DevoLearn platform, are these events that, you know, where the cells are moving around and they're dividing. The interest in these processes is taking place against the backdrop of genomic studies, particularly of global patterns of gene expression at the level of single cells. So people are interested in what the cells are expressing in terms of genes, what genes are expressed when, but that doesn't really account for the way cells build tissues and organs. So people have these very elaborate models of, like, they know the genome of C. elegans, they know some of the regulatory aspects of the genes, but then that doesn't tell you a lot about what's going on then at the cellular level, where you have migration and division. We know things about cell division in terms of, like, what genes are involved and how they're regulated, but we don't know how that actually maps onto, like, the type of real-time model that we're trying to build with DevoLearn. So there's a gap there. Here we discuss the need to integrate the activity of genes with that of cells, and propose the need to develop a framework based on cellular processes and cell interactions that parallels that which has been created for gene activity in the form of gene regulatory networks. So this is the GRNs. We begin to do this by suggesting elements for building cell regulatory networks, or CRNs. In the same manner that GRNs create schedules of gene expression. So they basically determine when the genes are turned on or turned off or turned on halfway or whatever, that result in the emergence of cell fates over time. CRNs create tissues and organs, space, in space. So they have this regulation of space, and they regulate, you know, what tissues and organs are built. So this is kind of almost like the differentiation trees. This is very similar in terms of the way it's conceived. We also suggest that GRNs and CRNs might interact in the building of embryos through feedback loops involving mechanics and tissue tectonics. So this is the sort of thing, if you've seen Susan's presentations where she talks about cell mechanics and development, some of the things that she's done, she's talked about, like this biological fracking that goes on in the embryo, or some of the, pressure, the role of pressure and the role of, like, other types of physical stimuli on cells. This is sort of the thing, this is the CRNs are getting at. The GRNs, of course, are affected by these things. So GRNs are triggered, for example, by stresses or by, other types of signals. There sometimes, if you have stresses like temperature or pressure, those can also have an effect on the GRN. It'll turn certain what they call early immediate genes on and then turn on other genes. But the CRNs are actually kind of filling this gap between what the GRNs are doing and the actual phenotype. And so they kind of go through an introduction here. And they kind of give, motivate the problem. So they talk a lot about, like, what's going on in the embryo in terms of different movements. So you have, we know that, like, a lot of developmental biologists are obsessed with like gene expression assays and looking at how different genes are expressed in the embryo. But then there are these other types of collective behaviors like invagination, convergent and extension, and cavitation. So these are things that happen at the sort of embryo level that are, when the cells when they reach a certain point, for example, when they're trying to build one of these germ layers and they're trying to, these germ layers are differentiating, there are these large-scale movements that happen like folding and bending and other types of displacements amongst the cells that, you can't really characterize with a GRN. And so this is what they're kind of trying to model, but they're doing it in a way that's, a regulatory network approach. So they're looking at these, so GRNs are a representation of gene expression. Their nodes are transcription factors and sometimes signals and signaling pathways. Their assembly can result in what they call circuits. So these are like brain circuits, except that they're on the molecular level. And these are small networks that perform specific operations, usually associated with motifs. And so these motifs are like sets of genes that are linked by their expression. So you can have things like coherent feed-forward loops, which provide temporal control and gene switching, and incoherent feed-forward loops that lead to oscillations and perform adaptation. So, if you couple some genes together and you control their expression, you can form these sort of control motifs, which allow you then to have like a, over time you can regulate what genes are expressed. And this helps you in development because there's this time dependency that you need to have. These circuits define programs of gene expression underlying cell fate decisions. And so the GRNs link to things like the Waddington landscape, which you've talked about a little bit. But then GRNs do not contain and cannot provide spatial information, such as volumes, geometry, densities, and the associated mechanochemical signaling. So this is where your CRNs come into play. And there are a lot of intrinsic cellular activities that you also need to model. And so these are things like integrating the activity of the cytoskeleton, the cell and cell matrix adhesion, and intracellular trafficking systems, which are actually the nodes of the CRN. So this is something that we've actually done with embryo networks, but we've not thought about it in terms of control and in terms of feedbacks. So this is something that is, kind of in our area of interest. So I thought I would kind of spend some time on it. Let me see if they have any figures in this paper. Okay, so these are the signals. This kind of gives you an idea of what happens in a cellular regulatory network. You have these chemical and mechanical signals from the cell environment. So these are GRNs here, these are CRNs. The GRNs focus on time, CRNs focus on space, although I wouldn't necessarily break it out that way, but and then figure 2, they only have 2 figures. They show this, I think this is an example of a cell regulatory network, and I don't know what these letters stand for, but they have, you can see what kind of a network they have. They have these things that, you have regulation in forms of these red arrows, and the blue arrows are the sort of connections between them. And you can see this thing where you have this crossover between the causal factors for CRNs and the causal factors for GRNs, and they affect one another. So that's what they're trying to point out. They don't really have anything implemented here, so that's something that is missing from this work, but I think it's just kind of proposing what these should look like. And so I don't know if you had any questions on that. I did want to talk about one more thing here, and that is this paper here, and this is a lighter paper. This is the abstractions blog from Quanta magazine. So this is a sort of a popular press article. This is where, let's see, ancient Turing patterns builds feathers, hair, and now shark skin. So a primordial developmental toolkit shared by all vertebrates and described by a theory of the mathematician Alan Turing sets the growth pattern for all types of skin structures. So this is a pattern in the This is a shark embryo, or a very early shark, and you can see that this whole cat-shark hatchling stained to reveal its skin teeth. So it has these skin teeth along the back here, and you can see this pattern. It's this dentical patterning, which looks like teeth, and you have two parallel rows near the dorsal fin. So this shows where this development of these skin teeth initiates along this midline section here, and then the denticles then spread out to dot the rest of the body over, as it matures, in keeping with the action of a Turing-like mechanism. So we've talked about Turing mechanisms before, and they're these mechanisms where you have a bunch of particles, and they're controlled by chemical gradients that control the expansion of different, they allow cells to differentiate in different ways, and they control the spatial extent of different cell populations. So, for example, these denticles are restricted at this point in development, and then later that constraint is relaxed and they spread out across the body. And there's a reason for this, and the reason is if you don't have this precise control, you can end up with all sorts of developmental mutations. In vertebrates, you have this mutation where it involves PAX6, which is a developmental gene. And if PAX6 isn't suppressed at the right time, you can end up with one eye instead of two. And they call that like a, it's the one, I can't remember the Cyclops. So they call it a Cyclops mutation. And so, this is just a matter of developmental gene expression going awry. And so this is why all of this is importantly coordinated in time. You can see this difference here where, you start off with cells in one place and then they expand given different chemical signals and, gene expression and so forth. So Alan Turing, besides, doing a lot of work for winning World War II and some of his other work in computer science, he also came up with this model of pattern formation. It basically outlines how endless varieties of stripes, spots, and scales can emerge from the interaction of two simple chemical agents or morphogens. So morphogens is just a theoretical term. And then, people have developed other types of theories, but this is one theory I think that is very interesting in terms of pattern formation. Really, in terms of pattern formation, hasn't been surpassed. The beauty of this work is that it shows that there might be a very strong conservation of this mechanism for forming anything from shark denticles to bird feathers. This study bolsters a growing theme in developmental biology that nature tends to invent something once and then plays variations on that theme. So we can use Turing, sort of tweaking the Turing model, this reaction diffusion mechanism, to come up with different types of phenotypes. We have these raw materials, and then we're just kind of reinventing how it's shaping. So these denticles, you can imagine in different types of shark, might develop in different ways. You might retain this sort of striping of the denticles, or you might allow it to spread all over the body, or there are different ways that these can be positioned based on the species or based on the ecological conditions or whatever. And so this can be controlled in development, and so that's what they're kind of getting at. And you can explain that using Turing patterns and Turing mechanisms. So this, Turing models excite developmental biologists because despite its simplicity, it can explain a lot of diverse patterns. Yet in practice, very few instances of patterning in nature have been definitively pinned to a Turing-like mechanism. So it's hard to prove. It's just easy to see that it's consistent with it, but it's hard to actually prove it and show, in a very rigorous way that this is actually what's going on. So you have all these different hair follicles in mice and feathers in chicks, and these are two different model systems that sort of are consistent with this. But again, we haven't proven that this is exactly what's going on. So yeah, this is really interesting. It goes on about sharks. It goes on about Turing mechanisms and development. So that's a really interesting paper as well. So I'm going to put these links in the chat, and I'm going to kind of wrap it up here. Do people have any questions or comments?
Are the tentacles of shark very hard, or they are also like feathers? Like feathers are supposed to be very, very soft, and so are the tentacles they are I actually don't know like how they are. So this is a question out of curiosity.
Yeah. I'm not sure. Like, let's see if they mention it here. Yeah, I'm not sure what they're actually like in terms of their material. But they have these. Yeah, they have these little. It's like an armor, so it must be hard. So
Yeah,
the
and I mean that's surprising, like, because feathers are soft and you know, and the denticles apparently are for different materials. So it's nice to see how diverse and how much variation can happen because of probably evolution or I don't know.
Yeah. Well, I mean, it's like you have this common mechanism, so you could have feathers or you could have scales, and they could be, they could be shaped in the same way. So you just use the same machinery in development and say, whether this organism has scales or whether it has feathers, and you have or hair, and it just, it allows for the patterning to occur. So.
Interesting.
Yeah. Anything else?
Nope, not from my side.
Okay. But Mainak or Jesse?
No, nothing from my side.
Okay, good.
Interesting papers, and it's nice to see the brain-bird vehicle stuff come around again as well earlier. But that's all for me, really. It's good to see everybody. I'm gonna have to set out now, but take care, everybody.
All right. Have a good